Okay, good morning. Today we'll be going through this question. It's a paper 3 question, and it is on a variety of topics as all paper 3 questions are on. But the main focus for the start is respiration, and afterwards we'll be talking about cancer as well. So firstly, we talk about this Otto Warburg found that cancer cells prefer to metabolize glucose by glycolysis. And this is contradictory to what we understand, and it's a paradoxical finding because we know that glycolysis is less efficient for producing ATP compared to oxidative phosphorylation. And this effect is known as the Warburg effect. So the first question, a very simple question, state why oxidative phosphorylation is more efficient at producing ATP than glycolysis. So it's a one-mark question, it's just stating facts. So oxidative phosphorylation produces 20 ATP molecules, while glycolysis only produces net 2 ATP. If you want to say glycolysis produces 4, it is also accepted. But it's net 2 because you have to remember the initial investment of 2 ATP at the start of glycolysis. So part 2, the mutation of your P53 gene was found to prevent expression of your SCO2 gene in your cancer cells. SCO2 codes for cytochrome C oxidase protein, which is the last electron carrier of your ETC. So they ask you to suggest and explain the effects of the lack of expression, and then another keyword here is the highly proliferative cancer cells. So the lack of your last electron carrier of your electron transport chain prevents transfer of electrons to your final electron acceptor, molecular oxygen. And because your electrons are not allowed to flow all the way to the end, the final electron acceptor, your electron carriers remain reduced, NAD+, FAD are not regenerated, and therefore they cannot participate in your 3 reactions prior to oxidative phosphorylation. And then, because they cannot participate, these processes cannot occur. So if these processes do not occur, and if oxidative phosphorylation does not occur, there is no proton gradient formed across your inner mitochondrial membrane. There's no proton motive force that can be generated. Chemiosmosis and oxphos cannot occur. Therefore, we rely now on anaerobic respiration. And because anaerobic respiration is relied upon, because they were asking for effects here, therefore, we talk about, so what if it's aerobic respiration? It will now lead to an increase in your lactate formation. Okay, next. Describe 2 possible consequences of the effects in part 2 on the survival of cancer cells. So these cancer cells are the same cancer cells that if you see above, they are, do it in red, proliferative. Okay, so they are multiplying at a very, very fast rate. Okay, so let's discuss the consequences. Okay, so firstly, the survival of cancer cells will decrease. Why is this? Because there is an increase in lactate concentration. And when there's an increase in lactate concentration, this leads to a fall in your pH because it becomes more acidic. Lactic acid is acidic. Decrease in pH. When there's a decrease in pH, your cellular enzymes are denatured, and when they're denatured, your cell metabolism is disrupted, and therefore cancer cells are less likely to survive. So because they ask for 2 possible consequences, and because this is a 4-mark question, you have to give the other side of the view, because the key word here is discuss. So, however, the blood vessels formed from angiogenesis can carry away the lactate as it is produced. Then pH is unaffected, and if pH is unaffected, your cell metabolism is not disrupted, and therefore survival is not affected. So for this question, it's kind of unique because you're talking about your two possible consequences from the above. So if at the start, if at part 2, you could not identify that there's an increase in lactate formation, you'll not be able to get the next 4 marks, which is kind of a pity, but sometimes you just get these kinds of papers. So you hope that you get nicer questions in the A levels. Okay, so we move on to part B. Part B, we're talking about aerobic glycolysis, which was the same thing as what we were talking about just now, the Warburg effect. So in this question, we also talk about AMPK. And AMPK plays a central role in regulating cellular energy homeostasis. And then we see the next protein, or the next gene also that we look at is the MIC gene. The MIC gene codes for the MIC protein, and the MIC proteins stimulate cell division in mice. So control mice in this experiment have artificially introduced MIC genes and normal alleles for the AMPK gene. So look at the table here. The control group, they have a normal alleles for your AMPK gene. Your group 1, they have 1 mutant allele of your AMPK gene. Group 2 has 2 mutant alleles of your AMPK gene. Okay. So we know that MIC genes are introduced into your mice. And because your MIC genes are introduced in your mice, they will all have an extra copy of your MIC gene. And the only difference between your groups here is the control group has normal alleles for your AMPK gene. Experimental group 1 has one that is working and one that is mutated. And those are the symbols there. And then for experimental group 2, we have 2 mutant ones. Okay? So that's why we have these symbols here. Okay, so let's label down on the graph so it's always easier to remember. So the square here is our control group. The circle here is our experimental group 1. The triangle here is the experimental group 2. So if we label it in the exam, it's easy for us to refer to it. And we don't have to refer to the table, then refer back to the graph, then table and graph, table and graph. We just look at the graph and we have all the information we need. Okay, so first question, explain why the control group of mice develop tumors. It's very simple because, as the question stated, they have an additional copy of your MIC gene. And because the question also stated that MIC genes lead to the expression of your MIC protein, and the MIC protein stimulates your cell division. Because there is an additional copy of your MIC gene, there is overexpression of your MIC protein, and hence it overstimulates cell division. And because of that, there is now the development of tumors. Okay, next. Very, very simple question. It's a trend kind of question. Describe. You don't even have to explain, you just need to describe. It's a 4-mark question. So, mice from group 2, this is your first point. Mice from group 2 displayed earlier tumor formation compared to group 1. And then you justify this with 50% of group 2 developing tumors at 7 weeks. Next, we talk about mice from group 1 develop tumors faster than the control group. There's no point in your second point to say that it developed tumors slower than group 2, because that is the exact same point you're making in your first one. Okay, there is no point in this one, no point in saying that it develops slower than group 2, because this would be the exact same point if you are making this point. So therefore, we're now comparing it to the control group. Okay, so mice from group 1 develop tumors faster than your control group. And to justify it again, we use 50% of group 1 developing tumors at 10 weeks. So why am I using the 50% of group 1 and 50% of group 2 developing tumors? So when you want to compare between your groups, when you're looking at your graph, you either hold your y-axis constant or your x-axis constant. What does that mean? So for example, we were talking about 50%. So if this was 50%, then we look and we trace down and we see where all these ones hit the x-axis. And we can do the exact same thing with our x-axis values. With our x-axis values, we can say that at 10 weeks, hold it constant at 10 weeks. And then you see for the control group, maybe it's about 60%. For group 1, maybe it's about 50%. For group 2, it's about somewhere between maybe 15%. Yeah, so when you compare your values, when they ask you to describe your differences in values using a graph, you can choose to either hold your x-axis values constant or your y-axis values constant. Okay, so last question here. Suggest possible explanations for the results in 1.2. Okay, so we know that the control group develops tumors the slowest. And for the control group, they have 2 copies of your functional alleles for your AMPK gene. So control group, slowest. Group 1 is the intermediate. Group 2 is the fastest. Then we link it to 2 plus, 1 plus, 1 minus, 2 minus. Going back to the genotype here, mentioned above. So because it has 2 functional alleles, the control group develops it slowest. When there's 1 functional, it develops at an intermediate rate. And when there's 0 functional alleles or 0 normal alleles, they develop tumors the fastest. So we can see a correlation between having the normal allele in your AMPK gene and having no normal alleles in your AMPK gene. The more that is present, the slower tumors form. So our functional AMPK, we give a reason why they develop tumors the slowest, because your functional AMPK inactivates the activator that upregulates your MIC gene. So this to explain why your development of tumors is slowest in your control group. Then now you say, when both alleles of your AMPK gene are mutated, now your AMPK is no longer functional. And when it's no longer functional, it no longer is able to inactivate your activator, your MIC gene remains overexpressed. Next, you talk about 1 allele being mutated. Because only 1 is mutated, there is still some functional AMPK present. And because there are some that are present, they are able to inhibit the activator, and this lowers the rate of expression of your MIC gene. So this sums it up for this question. It's a very typical paper 3 question. And yeah, I hope you've learned something from today. Thank you.